Content presented in Cheshire Corners is for educational and informational purposes only. Please consult a legal professional if you have any questions or concerns about the content of this disclaimer, this series, or the legal implications surrounding psychedelic substances in your jurisdiction.
All right, thanks everybody. We're going to talk about mushroom extracts. We're utilizing waste and extra parts of the plant, right? A lot of people trim their mushrooms and will cut the bottom of it off that has a little bit of substrate on there. And people are just throwing away thousands of pounds of this trim because it's got substrate on it. You don't know how to use it. If it's part of the mushroom and it's soluble, we want to pull everything out of it. This is a full spectrum extract.
Welcome to Cheshire Corners. I'm the AI clone of Rita Salyuk, your host, as we trip through the looking glass. And today, we're exploring the science behind mushroom extraction techniques. This video focuses on lab methods, chemical analysis, and mycology research, highlighting the fascinating biology and chemistry of fungi. Mushrooms are among the most chemically complex organisms studied in modern science. Neither plant nor animal, fungi occupy a unique biological category, producing a wide range of compounds that serve structural, nutritional, and defensive roles within their ecosystems. For researchers, this complexity presents both an opportunity and a challenge. Studying a mushroom as a whole can reveal broad patterns, but it can also obscure the roles played by individual components. To better understand how these compounds behave, scientists often rely on analytical techniques that allow them to examine specific parts of a complex system. One of the most widely used approaches is extraction. In scientific terms, extraction is not about making something stronger or more potent. It is about clarity. By separating compounds from a complex biological material, researchers can study chemical properties, interactions, and behaviors in a more controlled way. This approach is not unique to mushrooms. Extraction is used throughout food science, environmental testing, pharmacology, and materials research. In each case, the goal is the same: to reduce complexity so that meaningful observations can be made. When we talk about mushroom extracts in a research context, we are really talking about how scientists translate a living organism into data that can be measured, compared, and understood.
Unlocking the power of mushroom extract. We're about expanding safe community access through education, science, and support. Today, we're going to be talking about mushroom extracts, how they're made, how they can elevate safety, accessibility, quality.
Natural materials are inherently variable. Even mushrooms grown under similar conditions can differ in composition due to genetics, environment, and growth stage. This variability makes it difficult to compare results across experiments unless steps are taken to standardize what is being studied. Extraction helps address this challenge. By isolating specific components, researchers can reduce background noise and focus on defined variables. This makes it easier to repeat experiments, verify findings, and build on previous work. In scientific research, repeatability is essential. Results that cannot be reproduced are of limited value, regardless of how interesting they may seem. Extraction provides a pathway toward consistency, allowing researchers to compare samples across time, locations, and studies. This is why extraction plays such a central role in lab science. It is not a shortcut, but a tool for precision.
Okay, why extraction matters. Mushroom extraction is the process of separating the active compounds from the mushroom. We're going to prep our material, usually by drying it and grinding it up, reduce the particle size, increase the surface area, get good extraction efficiency. Then we're going to soak that material in some kind of a liquid solvent.
Before any analytical process can begin, samples must be prepared in a way that supports accurate measurement. This principle applies across scientific disciplines, from geology to molecular biology. Preparation is not about altering the material's identity, but about making it suitable for study. In the case of biological samples, preparation often focuses on improving consistency. Researchers aim to minimize variation caused by size, structure, or moisture content so that analytical methods can work as intended. Concepts like surface area, uniformity, and homogeneity are fundamental to lab science. They influence how materials interact with analytical tools and how reliably results can be interpreted. By approaching preparation thoughtfully, researchers create the conditions needed for meaningful analysis without introducing unnecessary variables.
There's the filtration process. That's where we're removing the solids away from the liquid. So there's additional refining steps to remove unwanted compounds or contaminations.
Once a sample has been processed, additional steps are often used to separate what is being studied from what is not. In scientific research, this process is best understood as quality control. Filtration and refinement allow researchers to remove unwanted material that could interfere with analysis. This might include insoluble matter, background compounds, or other elements that make results harder to interpret. Importantly, refinement does not imply that one outcome is better than another. Different studies require different levels of complexity. Some research benefits from broader mixtures, while other investigations require narrower focus. The key is intentionality. Scientists choose their approach based on the questions they are trying to answer.
Okay, so this is really important: full spectrum versus selective extraction. This is nothing new. This has been in the science world for a century. Full spectrum extraction, we're pulling all the soluble compounds out of the mushroom. If it's part of the mushroom and it's soluble, we want to pull everything out of it. This is a full spectrum extract. Selective extraction is when we're intentionally removing, or we're trying to avoid extracting, unwanted compounds from the mushroom. So things like sugars, lipids, fats.
The distinction between full spectrum and selective extraction reflects a broader theme in scientific inquiry: breadth versus specificity. Full spectrum approaches aim to capture as many soluble components as possible, preserving the complexity of the original material. This can be useful when researchers want to study interactions between compounds or understand overall composition. Selective approaches, by contrast, focus on narrowing the field. By emphasizing certain components and excluding others, researchers can examine specific variables in greater detail. Neither method is inherently superior. Each serves a different purpose. In fact, many research programs use both approaches at different stages, moving between broad observation and focused analysis as understanding deepens. This balance between complexity and control has been part of lab science for over a century, shaping how researchers design experiments and interpret results.
So there's lots of ways that we can decide whether or not we want to do a full spectrum extraction or selective extraction. So these chromatographs here are going to help kind of explain and show the difference between a full spectrum extract and a selective extract. So you can see in our full spectrum extract, it looks very similar. We've got the same peaks and the same positions that we see in the biomass, right? So the extract looks very similar to what we see in the biomass. Here's an example of an exotic fruit, just so you can see the difference. If you see these different peaks in here, this is why, it can be important to utilize a full spectrum extraction process so that you make sure that you're getting all these things that make it unique. We're utilizing waste and extra parts of the plant, right? A lot of people trim their mushrooms and will cut the bottom of it off that has a little bit of substrate on there. And people are just throwing away thousands of pounds of this trim because it's got substrate on it. You don't know how to use it. Utilizing waste, using every part of the fungi.
Modern research increasingly emphasizes efficiency and sustainability. In biological sciences, this often means finding ways to make use of entire samples rather than discarding portions that still contain valuable information. Using all parts of an organism can reduce waste, lower costs, and improve the overall efficiency of research programs. These considerations are especially important in large-scale studies and in fields connected to food systems and agriculture. Sustainability in science is not only about environmental impact, but also about responsible resource management. Efficient use of materials supports long-term research efforts and aligns scientific practice with broader societal goals.
And I hope you guys learned something about the extraction process.
Extraction is a powerful analytical tool, but it must always be understood within the context of responsible science. Methods, materials, and safety standards exist for a reason: to protect researchers and ensure the integrity of results. For audiences learning about scientific processes, it is important to distinguish between understanding concepts and attempting to apply them. Research techniques are developed and carried out in controlled environments by trained professionals. By focusing on how scientists think about extraction rather than how it is performed, we gain insight into the principles that guide modern research. Ultimately, the study of mushroom extracts is one example of a much larger scientific effort: the ongoing work of translating the complexity of nature into knowledge that can be shared, tested, and built upon. Thanks for watching. If you liked it, please like and subscribe to Cheshire Corners. Tell me what you think in the comments. And as always, don't get psyched out.